back, everybody. So this is my second video for protein synthesis. So make sure that you have watched the first part before coming here. The first part is talking about RNA and that first step of protein synthesis. And today we will be talking about the second part of protein synthesis. Okay, and before we start, I just wanted to show you this is the RNA strand that should be made from this template DNA strand, and this was from the previous video. This sequence was also included in the description of that video as well. But go ahead and take a look. And so just as a reminder, right, RNA, it works basically the same as the DNA in terms of what pairs with what. However, we do have, instead of T's, we have U's, right? So T stands for thymine, and in RNA we have uracil. And so where you would normally have a T pairing with an A, you would instead just have a U. Okay, so you'll see U's here. But other than that, it is the same. The G pairs with the C and vice versa. And if you had a T in the template strand of the DNA, then it would still pair with an A in the RNA. Okay, so double check that, okay, and then we can move forward. We're going to talk about today the next step of making proteins. All right, and so the first part of protein synthesis that we talked about in the previous video was called transcription, right? And so we talked about how DNA is stored in the nucleus, right? It's double-stranded, it's very long. All right, so we store it in the nucleus, and we, instead of taking the DNA out and using it directly, we actually have this intermediary step of the mRNA, right, the messenger RNA, and it's smaller, right, it's single-stranded. We can do one gene at a time, and it will leave the nucleus, and it will go to the ribosome, and then it can then be read by the ribosome as a code to make proteins, okay, right, so it leaves the nucleus, it goes to the ribosome, and this process that we're now going to talk about is called translation. Okay, and so if you think about, we were first transcribing DNA to RNA, right? The code is basically staying the same, but then we're not translating it, like turning it into a different language, and the language is actually going to be the proteins, and that is what will then control your traits. Okay, so let's see what happens. All right, so translation, ultimately what happens is we have a ribosome, it reads the mRNA, the messenger RNA, and it's going to create a chain of amino acids, right? So the amino acids are the building block for the protein, okay, which is our end goal here. And it's going to happen right in the ribosome. So we could be out in the cytoplasm, or we could be on the endoplasmic reticulum, okay? So we can see here, this is showing the mRNA is coming out of the nucleus, which is this purple ball here, okay? And it has, you know, its little letters on it. You can see there's AUG, UUC, AAA, okay? And then the ribosome is going to read it, and it's going to put together the amino acids. You can see here we're ending up with what is called a polypeptide chain. Poly for many. Peptide refers to the bonds that's holding the amino acids together. So we can call it a polypeptide chain. All right, and if you zoom in here, you can see here we've got different amino acids, and we will discuss that in a bit. All right, and so you might be wondering, well, how does the ribosome know which amino acids to put together? Well, mRNA, messenger RNA, it's a code, and it's read like a code, much like, you know, a language or a computer program or something like that. And it's pretty simple. We read them in sets of 3 bases, so 3 letters at a time, and we call that a codon, okay? So it's a set of 3 RNA bases, and it codes for a specific amino acid. So you can see here in my little picture, you have the RNA, so it's a single-stranded spiral, and we can divvy it up into these sets of 3 letters, and each of them is going to be a codon, okay? So we have 7 codons here, so we have 21 letters in total. Okay, and so like I said, right, each codon that codes for a specific amino acid. So if we go back to this picture, each of these little colored dots that represents an amino acid, it has a 3-letter word essentially that codes for it, okay, and that's what the ribosome is reading. We also have special codons, okay, and they code for the beginning and the end of the protein. So that way the ribosome knows, you know, where does it start, where does it stop, Okay, and so we have special ones here, and it's good to just be familiar with what they are. So AUG codes for the beginning of a protein, and that will always code for the beginning. Okay, so whenever you see an AUG, then you know that's where the protein starts. And then stop codons, there's three different ones. And so any of these three can tell the ribosome, hey, we've gotten to the end of the protein, we can stop here. All right. So ultimately, codons are like three-letter words. Okay, we've got plenty of those in the English language. I've got some examples here: cat, hat, bat, big, wig, not, hot, lot. Right? You can spell a lot of different words with just three letters. And similarly, with the three letters we have for codons, we can have a specific amino acid that it codes for. All right, and so this is just showing, right? It's kind of like a code. Okay. And so once you learn how to read it, then you can see exactly what that messenger RNA, what kind of protein it's going to make. Okay. All right, and so once we have this code, then we can think about putting everything together, right? So this is when we're starting to think about the tRNA that I mentioned in the previous video. So the ribosome is reading the mRNA, okay, the messenger RNA. Now the tRNA, which the T stands for transfer, it's going to attach the correct amino acid. So the tRNAs are actually they're kind of floating around the cell in the cytoplasm, and they're all holding an amino acid at the end here. So you can see here, this is one way to represent a tRNA, and we have an amino acid attachment site here. And it's going to be holding an amino acid, okay? And depending on the type of tRNA, it's going to be holding a specific amino acid. And the way that we can tell which amino acid is holding is the tRNA itself also has a little code, okay? And we call this the anti-codon because it's supposed to match to the codon on the mRNA. So look at this picture here, okay? And so at the very bottom of this tRNA, we have the anti-codon. And you can see here, it's going to match up letter by letter to the codon on the mRNA. Okay, so if the mRNA codon is GAG, then the corresponding anti-codon will be CUC on the tRNA. So the tRNA is going to go essentially find, the codon that it is matched up to, okay? And then that is how the ribosome can then put the amino acids in the correct order and then bind them together. Okay, here is just another representation of the tRNA. Okay, here it's been simplified into kind of rectangle shape, but it's carrying this amino acid. All right, and this is actually the amino acid that starts the protein, right? So AUG is our start codon. And then the anti-codon will be UAC. And so the tRNA comes in, sticks to the codon that it matches to, and then you can imagine them all kind of sticking in there, and then we can bind the amino acids together in the correct order. All right, here's just a sort of overview picture of what's going on, right? So we can start on the left here in the nucleus. So the DNA here, we have, one chromosome all wound up. It's one DNA molecule. And then we have one RNA, and this should be mRNA, to represent, one gene that codes for one protein. It can then leave the nucleus. It's going to go, see now it's mRNA. It's going to go to the ribosome. Okay, and the ribosome itself is made up of RNA, right? It's made up of ribosomal RNA. It's going to put together the amino acid chain that's coded for by these codons, right? With the assistance of these tRNAs that are holding the amino acid. They're going to come into the ribosome. So you can think of it as a little protein factory. The amino acids will come in. All right, they're going to bind to the correct codon with their anti-codon. All right, the amino acids will be bound together by the ribosome. And then once the tRNA has come in and given its amino acid to the growing chain, then it can go pick up more amino acids. Okay, so you can think of the tRNA are these little factory workers that are, bringing in the materials and then going off to get more. So you might be wondering, all right, well, how do we know which amino acid is coded for by which codon? And so this is not something that I would expect you to memorize. And in fact, that's why we have these charts and wheels that you see on the screen right now. And they're a way for us to basically decode, all right, the messenger RNA. Okay, so let's just take a look, right? So we have 3 letters in our codon. I'm going to start with the chart here on the left first. And so the way that you can read this is we have the first letter on this side, okay, and then we have the second letter along here, and then you can look in the individual boxes for the third letter. But let's say, right, our start codon, if you remember, is AUG. Okay, so we'd start here, and that's the first letter is A. Okay, and then we want to look along here for the second letter. The second letter is U. So we are in the third row in the first column. And let's look in here. Now we've got AU and then G here. So that codes for start. And that is the amino acid MET, which can be represented with an M. Its full name is methionine. Okay, and so that is how you would read this chart to figure out, what does this code actually mean? It's almost like a dictionary, right? So you look in the dictionary, you figure out, how is this word spelled so that you can then look it up in the dictionary to see what it means. It's the same exact thing, except honestly, it's much more simple than the dictionary. Okay, we don't have nearly as many amino acids as we do words in the English language, thank goodness. But let's take a look at the wheel, okay? The wheel to me is a little bit more intuitive, but I do have both in here just depending on your own personal preference. But how we read the wheel is we start in the center, and we'll use AUG again as our example. Okay, we'll see A is in this quadrant of the wheel, right? So you can see here in the inner part of the wheel, it's split into 4 quadrants. So we'll start with the A. So then now that we've chosen our first letter, we want to be limited within this quadrant, right? So then we see, okay, what's the next letter out of these 4 is U, right? A-U. And then the third ring of the wheel, right, would be our third letter. So G here. So you can see A-U-G, that codes for methionine. And this wheel has the full names of the amino acids. Okay, and so you can take a look, all right? So we have a number of amino acids. And so, in fact, the number of amino acids that we have that our body uses is 20. And some of them your body can make on their own, and some of them you actually need to consume through food, which is part of the reason why you need to eat. But we have 20, and so having these 3 letters to code for 20 would actually have a lot of overlap, okay? So let's take a look. How many different codons code for the amino acid GLI, all right, which is glycine, okay? So you can take a look either on the chart or the wheel, but we're looking for glycine, right? Okay, so here is glycine, and if we work backwards, we can see there's actually GGG codes for glycine, GGA codes for glycine, GGC codes for glycine, and so does GGU. All right, and I think on there, that's all the ones that code for glycine. We can see there's 4 different codons that code for glycine. And so we have what we call redundancy in the amino acid code. And you can take a look at that is true of all the other amino acids. You can see serine has a bunch here that it's coded for. Leucine has a few here, but leucine also has more here, you'll notice. So CCU, CCC, CCA, CUG. I think I just said that wrong. Anyway, all the ones that have CU and then have any third letter code for leucine. And then we have UUA, UUG. All right, the point being, okay, that the amino acid, one single amino acid has a lot of different codons that will code for it. So it's almost like there's a bunch of different words and they all have different spellings, but they all mean generally the same thing. So it's like synonyms if you want to use the dictionary analogy. Okay, and so the redundancy, ultimately what it does for us is that once we get to talking about mutations, right, it tries to minimize how damaging the mutations are, right? So we'll talk about this more in a separate lecture. But just keep in mind that we have a bunch of different codons that can code for the same amino acid, and that is ultimately a good thing for us. All right. And so I will post, I believe this is the end of my lecture. Yeah. I will post some practice because the transcribing and translating, I think, is pretty straightforward once you get the hang of it. But it is one of those things that is helpful to practice. So I would give you, some sequences and see if you can translate them for me. Okay, but that is really the end of my protein synthesis lecture. So we've gone from DNA in the nucleus to mRNA, which can leave the nucleus and go out to the ribosome. All right, and the ribosome, with the help of our transfer RNAs, can then put together this amino acid protein sequence based on the code that is in the messenger RNA. Okay, and so that is our central dogma, okay, of going from genes to proteins and then ultimately to traits, right? So, and we'll talk more about the different things that proteins can do. But that concludes my protein synthesis lecture. I hope it was clear and helpful, and I do welcome any feedback. This is all quite new to me, doing this kind of video recording. And so I would love to hear what you think and how I can improve this so that it's more educational for you. Okay, but I will hopefully keep on doing this so that we can at least have the opportunity to continue with the curriculum for those of you who are interested. And so I will see you hopefully in the next video. We'll talk more about genetics in the next one. All right, so bye-bye. I hope you have a wonderful day.